Discover a mechanism of immune response which also affects the behavior of cancer cells. Explore real-time rendering of scientific data. And glimpse what the future may hold for safer driving environments on this episode of OnBeyond.
In a discovery that has implications for controlling the spread of cancer, UCSD biologists have unraveled the mystery of how cells orient themselves. Their discovery of the genetic and biochemical mechanisms controlling this internal compass may also limit the pain and swelling associated with arthritis. The finding explains how white blood cells are guided with missile-like precision to the sites of infections. It also explains the fundamental mechanism by which other cells are able to find nutrients and hone in on other chemical cues within their fluid-filled environment. UCSD biology professor Rick Fertel explains.
What we have discovered is the internal compass that allows cells to move to a directional signal. To give you an example of this, if you have a wound that's infected with bacteria, various types of white blood cells will rapidly move to the site of the infection, attracted by the bacteria. And the goal of the white blood cells is to kill and to eat the bacteria, cleansing the wound. And this is the body's first attempt or mechanism to fight outside infection. Another example is if you have a site of inflammation. Inflammation is often caused by small amounts of tissue damage. White blood cells need to move into the area and essentially clean out the damaged tissue. So the cells need to be able to figure out where they need to move to, how they need to find the source. And this internal compass that I mentioned allows cells to localize the site. The bacteria themselves produce small molecules, chemicals, that are recognized by the white blood cells. And the internal compass gives the directionality of that signal. And it tells the cells where those small molecules are coming from, and therefore where the bacteria are in the body. The difficulty of using, for example, real white blood cells for this type of analysis is the absence of the ability to do real genetics, to be able to identify mutations within a pathway. Genetics is one of the key basic tools for discovery in biology. By making mutations that affect the ability of cells to, for example, move towards bacteria, we're able to identify new genes that are involved in this pathway, and thus able to uncover the actual mechanisms that control this process. The organism that we use for this type of analysis is Dictyostelium discordium. Dictyostelium is a soil amoeba that has many of the same properties as white blood cells. They are attracted to bacteria in the same way that white blood cells are attracted to bacteria, and they're able to eat and destroy bacteria. We have made use of Dictyostelium in order to define what this internal compass is. One of the other advantages of using Dictyostelium is that these cells move very vigorously and very rapidly towards one of these chemical sources. This provides a significant amount of advantage to us. Number one, we can do recordings and analyses in real time over a relatively short period of time, 10 or 20 minutes. In contrast, to get the same type of analyses on white blood cells, it may take an hour or 2 hours or 10 hours in some cases. Furthermore, if we want to look at mutations that have an impact on the ability of the cells to move, and for example, let's say the cells move 50% slower, half the speed of normal cells, of wild-type cells. If we went and did the same type of mutation in white blood cells, it would almost take forever to do the analysis, and the quantitation would not be as accurate. So by using Dictyostelium, we can get quantitative numbers and therefore really understand what the different genes, what the different proteins are doing in controlling this process. This is a micropipette that contains the chemoattractant source. And the dictyostelium cell is moving directionally towards the micropipette. And what we can see, you get a sense of this compass. By moving the micropipette, the cell now is able to change its direction. It's able to put out a new front of the cell and essentially move now in the new direction of the pipette. We can also see that the cell, when it starts moving, becomes elongated, or what we call is polarized. And you can actually distinguish, visualize differences between the front of the cell, which has the motor machinery to essentially move the cell forward, while the back of the cell, which has a different motor that allows the back of the cell to contract and essentially follow, get to the front. This type of directional cell movement has a very, very important applications in various aspects of medicine. I talked about white blood cells moving to the wound. During our normal body development, cells need to move from various parts, from one part of the body, developing embryo, to another part of the embryo, and it again utilizes the same mechanism. This is also very important in cancer. As we all know, cancer often starts at a particular location in the body and metastasizes, or basically moves out of this area and forms new sites of infection in other parts of the body. And the spreading of the cancer is quite often which is what kills people. Breast cancer cells, for example, initially just are localized in a relatively small area of the breast, but they then gain the ability to move out or to metastasize into the bloodstream. And it's now understood that one of the key mechanisms that mediates this movement into the blood vessels is by this directional movement. And cancer cells acquire the ability to recognize similar types of small molecules that are produced in various parts of the body. For example, in the site of infection, these small molecules are found in blood vessels. They permeate the body, and what happens is that the cancer cells, the breast cancer cells, recognize these signals and utilize the same internal compass, the same directional movement, to now move out of the site of infection into the breast, into the blood vessels, into the lymphatic system, where it then can essentially infect other parts of the body, leading to a significant illness and death.
Biologists in Fertel's UCSD laboratory, Satoru Funamoto, Rudy Melli, Susan Lee, and Lisa Perry, first determined that movement toward chemical attractants in dictyostelium was controlled by two genes. One produces the protein PI3 kinase. The other produces the protein PTEN, which is associated with half of all human tumors.
The discovery that we made identifies two genes that in fact have already been known and have been under intense research in a number of laboratories, including a number of laboratories at UCSD. These genes regulate cell growth. So they've been known for a significant period of time of controlling cell growth, and themselves have been associated with a large number of cancers. One of these is called PI3 kinase, and what it does is it adds a phosphate group onto a lipid in cell. The other gene is called PTEN, literally PTEN. And PTEN, and one of the discoverers of PTEN is Webb Cavany at UCSD. And PTEN is the negative regulator of PI3 kinase. And the way to think about this is PI3 kinase adds this phosphate group to the lipid, and PTEN removes the phosphate group. Now, what does the phosphate group do when it's added to the lipid? The phosphate group added to the lipid is actually part of the internal compass. That provides the mechanism by which certain other protein molecules localize at the front of the cell, the part of the cell that's essentially going to be in the driver's seat that allows the cells to move to, for example, the site of infection, or allows the cell to move out of a tumor. So PI3 kinase initiates this pathway, and under normal conditions, PTEN basically eliminates the pathway. It basically turns it off. Now, PTEN has been identified in more than 50% of human tumors, which clearly indicates its very, very major importance in human disease. What we've discovered is that PI3 kinase is essentially the driver in controlling this movement, and PTEN functions as the brakes. Now, it both functions as the brakes to turn off the pathway if, for example, the signal is no longer perceived, but it does something else very important. If we think about a compass and we want to move in the direction, we keep the needle pointed in the direction that we want to go. If we're in the driver's seat in a car, we're trying to hike, and if the needle is bouncing all around, and it first points to north, and then it points to east, and then it points to west, and it's bouncing all around in these various directions, we essentially have lost the ability to sense direction. We don't know where to move. What we found is that PTEN helps restrict or localize the compass. For example, if the PI3 kinase pathway wants to point north, the cell needs PTEN to keep the compass pointed in the right direction. If you remove PTEN from the cell, then what happens, PI3 kinase says, well, I sort of sense the right direction, but it's bouncing around between north and east and west. So PTEN sort of restricts and localizes the signal to the right part of the cell. And in that regard, it plays an absolutely essential role. Now, I think what has maybe stunned people, maybe is an appropriate way of putting it, is that these components that have been so important in controlling cancer now control cell movement, something that actually evolutionarily is much older than cancer, much older than the ability of mammalian cells to control their growth. So we can think about it in a slightly different way, that directional movement, the internal compass, evolutionarily is much older, and as mammals evolved and as humans evolved, we essentially have usurped this pathway to now control the ability of cells to grow. So this pathway controls growth. If you remove PTEN, you now are very, very prone to a large number of cancers. And if you remove PTEN from the ability of cells to move directionally, then they essentially have lost their signal, have lost their ability to move in the right direction. We had discovered PI3 kinase in dictyostelium several years ago. And because of its key importance in understanding cancer and understanding human cell growth, we wanted to pursue and try to understand how it may function in this organism. And what we discovered is by making a mutation in the genes encoding the various PI3 kinases, we essentially found that these cells were no longer able to move effectively towards one of these chemical sources. What we then did was try to understand how PI3 kinase was regulated. And in that way, we discovered PTEN. And when we made a mutation that eliminated PTEN from cells, what we found is that the cells still had a compass, but the compass could not focus in on the appropriate signal. What we also discovered is that if we now produce more of PTEN in the cells, you basically lost the ability of cells to move because they've lost the internal compass. So in reality, PI3K, PI3 kinase, and PTEN are essentially the yin and the yang of this process. One activating the process, and the other one functioning in two ways. One is to control the process so it doesn't get out of hand, and the other one is to focus and restrict the process so it only occurs at the very, very front of the cell.
When a chemoattractant is added to the fluid medium, normal or wild-type cells move rapidly and directly toward the source. By contrast, mutant cells without normal amounts of PI3 kinase produce pseudopods in multiple directions as if they are unsure of which direction to go. The cells also move significantly more slowly.
One also sees that the cells are still able to move towards the micropipette. And we believe this is because there are in fact 3 of these PI3 kinases in this organism. And we have essentially genetically knocked out the major ones, PI3K and PI3K1 and PI3K2. But there's a third one, PI3K3. And we think that the remaining residual information that the cell is receiving is due to this additional PI3K3. Now, in addition, the back of the cell sort of becomes stretched out, elongated, and it's not the same as the normal cells. If you look at these cells, essentially they're dragging their backside. And the PI3 kinase pathway, this PI3K pathway, not only controls the ability of the front of the cell to move, but also controls the ability of the back of the cell to retract. So both parts, the front and the back, are regulated by this pathway. And you think of it like an inchworm. The front moves forward, the back then follows it. And it's this general basic mechanism that controls how all individual cells move. PI3 kinase, we knew, plays an absolutely essential, critical role in cell movement. We knew that if you remove PI3 kinase from the cell, if we made a mutation of PI3 kinase, the cells lost their ability to move directionally and move much more poorly and could not essentially elongate. But we didn't understand how this directionality was mediated. I mean, how does PI3 kinase actually function as a compass? The discovery of this came when we started actually looking at where PI3 kinase is in the cell. Now, the tools that we utilize for this is to tag the protein, tag PI3 kinase, it's a protein, with a fluorescent marker. This fluorescent marker is called green fluorescent protein, and it's an actual fluorescent protein from a jellyfish. What we can do is make a fusion of combining PI3 kinase with this GFP, this green fluorescent protein. So they are now a single protein in cell. Because green fluorescent protein is fluorescent, we are now able to visualize it using ultraviolet light. And then we need to filter the UV light such that we only see the fluorescent image. So what you're looking at is only the green fluorescent protein being illuminated by the UV light. And again, as we move the micropipette, you can see the green fluorescent protein, the PI3 kinase, move from one side of the cell to the other. Now, what we've been able to do subsequently is to try to understand what parts of the PI3 kinase actually control this movement. And through these studies, we hope to really be able to understand what now is the next step in the process. How does the PI3 kinase actually find the front? In addition to looking at where PI3 kinase is in a moving cell, that it's at the front of the cell and it basically functions as a compass, we've also looked at where PTEN is in the cell. And this also gave an extremely exciting finding. If you think about the cell as an oval, PI3 kinase is at the front, and it's sort of like the beginning top crescent of the oval. PTEN is actually found on the edge of the cell, but on the remaining part of the cell. So it's missing at the very front, but it's on the sides and the back of the cell. And the reason why this is so important is that if we think of PTEN as turning off the pathway, think about a horse and a buggy with blinders on a horse. The idea of the blinders is to keep the horse looking forward, looking in the right direction. So PI3 kinase needs a little bit of help. It's on the membrane, but what we want to be able to do is keep it focused at the very front of the cell. And what PTEN does by being on the sides of the cell is it keeps PTEN off. So we have PI3 kinase at the front and PTEN functionally as a blinders, focusing where PTEN is directed and therefore functioning to keep the compass pointing in the right direction. I think one of the most surprising discoveries is that cancer genes also control directional cell movement. Molecule PI3 kinase, which is at the head of the pathway that often leads to cancer, and many of the components downstream from PI3 kinase are known oncogenes, genes that are known to control cancer. PTEN basically puts the brakes on cells. And removing PTEN, the cell's growth becomes uncontrolled and they become cancerous. I think it's fascinating in biology that you can take PI3 kinase and PTEN that can control cell movement, that they're also critical for controlling proper cell growth. And if you remove PTEN, you get cells that are unable to properly sense direction, and you also get cells that are now oncogenic, that are now basically cancerous and will metastasize. Probably the absence of PTEN in these cells may also be one of the reasons why the cells become so motile and why the cells are able to move out of a cancerous growth. We believe these discoveries are going to have strong potential in possibly treating various types of cancer. If we think about breast cancer, there are two aspects. One is that we want to prevent the spreading of the cancer, and then we want to eliminate the cancerous growth. The identification of PI3 kinase as a key and critical component in directional movement and possibly metastasis allows one to develop drugs that could be targeted to the site of a tumor that would prevent the tumor cells from metastasizing, basically spreading into the blood vessels, into lymph nodes, into other parts of the body. One could then combine that with other types of treatments such as chemotherapy or radiation treatment, localized radiation treatment, which focuses in on the particular site of the cancer, essentially destroying the cells. So essentially we want to have a two-stage type of approach. One is to prevent the spreading of the cancer by preventing the cells from moving, and the other is to actually destroy the cancer cells. And we think that while the cancer cells are being destroyed, preventing the metastasis is a really excellent mechanism of ensuring that the cancer in fact does not spread anymore during the actual treatment process. Another possible use is the treatment of sort of mild arthritis. Mild arthritis is an inflammation, and again you have cells moving towards the site leading to people having chronic pain. One possible approach might also be not only to have intervention at the site of the pain, but also to have intervention to prevent the white blood cells from actually moving into the area. So by targeting against arthritis multiply at various different stages in the process, it's possible that some people that are not effectively treated with the present drugs might have other mechanism of treatment and might be relieved from some of the pain that this disease causes.
We're here in the visualization lab of the San Diego Supercomputer Center, and today we're going to look at ways of looking inside three-dimensional medical data. Let me show you an example of what I mean. Now, of course, everybody is very familiar with three-dimensional graphics. We all play video games, we all go see movies. Typically in a video game or even in movies, three-dimensional things are defined in terms of very small flat surfaces called polygons. And here we've brought up some medical data. Here's a human head. And it looks very much like it's made of polygons. This is sort of the video game view of things, as you can move things around and see what they look like from different sides. But in fact, this is different. This is something called a volume. And a volume isn't a bunch of little flat surfaces. A volume is a whole cloud of 3D points. Oftentimes this happens as a result of getting a medical scan, because a medical scan consists of a number of slices, and all the little picture dots on each slice, when you stack them all against each other, becomes a cloud of points. And that's really what this is. If this was just a polygon model, that is a lot of the little flat surfaces on the outside, yes, we could move it around like this, but we couldn't go see inside it. And seeing inside things is a really important part of our research. So for instance, I can go over here to user interface. I can come over here and grab the transparency knob and lower the transparency. And as I do that, it's making the outside more transparent. And as you'd expect, the brain starts to show up on the inside. This really lets us peek inside things, which is important to us. How it does this is kind of interesting. Again, if things are made of polygons, those little flat surfaces, it just tries to draw the flat surfaces. But here, because it's a cloud of points, it can't really draw the cloud. Instead, what it does is it starts at the back of the cloud and starts working its way forward. And every time it encounters one of these three-dimensional dots, it blends it. It takes its color, it takes its transparency, and blends it into what it already has. By the time it gets done coming all the way through the volume, we end up with the image that we see. So every time we change something here on the screen, it's actually ray tracing, retracing through all of the voxels that make up this image. Well, now the fun begins. Some things we like to do with this right away is we like to first of all adjust where the light is. The light is important to us because depending on how the light shines on the object, it tends to bring out certain features. We've been really surprised how much information you get by moving this light source around. Some other ways of doing volume visualization don't really allow this kind of light source movement, and we've been very surprised how much more that gives us. Let's go peek inside. The thing I really enjoy doing with data like this is stripping away part of it. So let's come down from the top and we'll just start stripping our way through the data. Again, the system is retracing this back to front every time we change something here on the screen. So now as we move this slider back and forth, we're seeing through the top of someone's head. And in fact, if we want, we can change the bottom as well and really make a whole slice, and now we can move top and bottom together and really check out what's going on in this particular set of data. It also is very instructional to come in from the side and do the same thing. I like this view a lot because you can really see the center part of the brain, you can see the brainstem and so on. Also, looking at it from front to back is kind of instructional as well, because you can really see where things like the eye sockets are, you can see the optic nerve as it connects to the brain. So scientists use this kind of tool all the time in an effort to understand the insides of things. And medical data is probably one of the best examples of this. I also wanted to show you that the volume work that we do here also extends beyond medical images, because a lot of scientific data ends up as volumes, even though it didn't come from, say, a medical scanner. This is some work we're doing with Bernie Jackson, who's here at the UCSD Center for Astrophysics and Space Sciences, and one of the things he does is study the solar wind. Well, the solar wind is really intensity of the field of the sun in space, so it's a 3D volume. And so we can take his data and we can display it. Now the neat thing about this really is a cloud. And so if we adjust the transparency like we did before, we can really make this look wispy like the cloud. Although because we can also adjust the light source, we can really move around and understand the true shape of this data. I have one more volume I want to show you again, just to show you the range of things that can be done with this kind of technology. This is a submarine. In the Civil War, the Confederacy launched a submarine called the Hunley. And the Hunley was involved in a couple of battles in the Civil War, and then it sunk. It was recently raised by a group on the East Coast called Friends of the Hunley, and one of the first things they did was scan it. You can buy big 3D scanners, which will then go around a 3D object scanning for XYZ points in 3 dimensions. Unfortunately, then it's hard to look at. That is, it doesn't give you polygons, it doesn't give you lines, it gives you a cloud of points. Well, a cloud of points, that suggests that the kind of technology we use for medical graphics can be brought to bear on this as well. And so, in fact, that's what happened, is they sent us 9 million 3-dimensional points defining the submarine, and we brought it up on this program. And it looked like this, but once we got playing with the light source, we found that we could really bring out interesting detail. And so, in fact, their 3-dimensional scans did tell us an awful lot about the nature of what the Hunley design was. And so by using this exact same tool that we've been using for medical graphics, we've been able to examine a lot of different surfaces as well. Here's another data set that I think everyone really enjoys looking at. There's a project in the National Library of Medicine called the Visible Human Project, and they scanned someone who willed their body to science, and they scanned that person at great resolution. And so we've taken some of that data here and allowed you to look around in it. This one's kind of fun because it's real easy in this data set to strip away the outside and be left just with the inside. And that's pretty dramatic. We see here the skull, we see the brain. Well, as you've seen, we can use this kind of technology to see both the inside and the outside. If we leave the transparency low, then we can see just the outside. That's the first thing we come to. If we make the transparency a higher value, we can see through it into the inside. But what's neat is sometimes you want to see both of them together, and it's hard to do that with just the transparency alone. So to do that, we end up playing a little visualization trick known as the magic lens. What we'll do is we'll make it very opaque, and we'll say, remember that. Let's just simply get rid of the outside, and we'll get to this view, and we'll say, remember that too. And now we'll use a technique known as the magic lens. In the magic lens, we're displaying the first view outside this little box, and we're displaying the second view inside the little box. And notice that as we move around, we're actually seeing both. That is, we can really look at context. Here we see the jaw in the skeletal view, here we see the jaw in the skin view. And we can go back and forth like that very nicely. The next thing I wanted to show you is another interesting application of 3-dimensional medical graphics. This also comes out of the UCSD Medical School. We're working with a researcher named Tom Nelson, and his research area is to do 3D ultrasounds of pregnant women. Now, we all think of ultrasound as being something very fuzzy and hard to look at, but in fact, lately the ultrasound technology has gotten dramatically better. And in fact, not only can they see very clear images that even the rest of us can understand, but they can put them together into a 3-dimensional volume. Well, what happens is they then send the volume to us, and we visualize it here, and we also end up making physical models of it as well. So this is a recent data set where they had a set of twins, and as you can see, the detail is very impressive, particularly if you've seen ultrasound from a few years ago and know what fuzzy used to look like. But anyway, right down to the detail of the umbilical cord, the sucking of the fingers. Here, this one seems to be holding his umbilical cord. And so being able to actually see these 3D ultrasounds, being able to see what's going on in the womb, really spells a lot of good benefits in the future for fetal development, for understanding fetal development. One of the things that's really exciting about this is that it's so new. It really was not very long ago that all of us avoided volume-type visualization because it was so slow. Even with a supercomputer center, you couldn't really interact with it. You had to push a button and wait at least a few seconds to get one view of it. The idea of grabbing it with a mouse and rotating it was completely out of the question. Technology has changed, and our software has followed the technology so that we can do this. This whole setup that we use for this kind of visualization has been created here at the Supercomputer Center. We're using a commercial board called the Volume Pro card, which we plug into a PC that's sitting here at my feet. We're using some software that was developed here at the Supercomputer Center called the Volume Explorer. And if you combine all of the things that we have right here, it's really a desktop system. The total cost would be about $4,000 to put that sort of thing together. Well, one of the things that we hope is by having this in such a nice desktop form is that rather than having a situation like we are now where you bring data into our visualization lab and use it here, what we want to do is push the technology out to where the data is being gathered. And so one of our projects in the future will be to get this technology into medical scanning labs, 3D ultrasound labs, and so on, so that as patients are being scanned, doctors right then and there can bring up the three-dimensional display of it, interact with it like we've been doing here, and then possibly making some changes, rescanning the patient, making some decisions right there on the spot. The more that you can get the doctor and the visualization in the loop, we think the better the diagnoses will be. So despite the fact that we're really a supercomputer center, SDSC has been very good about doing other connected technology around the supercomputer that doesn't necessarily even have to use the supercomputer. And so this is a very good example of that.
The merge of Interstates 5 and 805 near La Jolla. It's one of the most congested stretches of freeway in Southern California and one of the most likely to be crippled by traffic accidents. Yet a stone's throw from the intersection on the campus of UC San Diego, several research projects are now underway to harness technology for safer roads, safer cars, and safer drivers.
They started explaining what were their problems, and then we started figuring out if there is anything that we can do given the technologies we had.
That's Mohan Trivedi talking about auto safety officials and industry executives who turned to him and other researchers in search of innovative technology to address auto safety and traffic congestion on American roads. Trivedi is a professor of electrical and computer engineering at UCSD's Jacobs School of Engineering and director of the university's Computer Vision and Robotics Research Lab. It is in this lab that Trivedi and his team are actively working on auto safety projects that are based on a very simple idea: computer vision, cameras in the car and along the road to monitor the state of the driver and the state of the driving environment. Computer vision, according to San Diego-based automotive technology consultant Erwin Boer,
can monitor what the driver is doing. It can perhaps monitor where the driver's eyes are. If you know that the driver is looking at a display 3 seconds, glancing at the road for half a second, then looking at a display again for 4 seconds, the potential that this driver will miss something that is happening outside is much greater. So it's basically twofold. It can establish a map of the environment to place the car in the context of the environment, as well as assess the state of the driver.
Trivedi's work on auto safety grew out of research commissioned by the California Department of Transportation, Caltrans, to find a way to cut down on the typical 40 to 50 minutes it takes to relieve congestion on California freeways once it builds up. Congestion that costs the state an estimated $2 billion a year in lost productivity, higher gasoline bills, and pollution.
We found out that there were significant savings that can be accomplished if, say, we had large area video camera coverage, and these were smart cameras, so to say, so they will be able to analyze traffic patterns, and even before an incident has happened, we might be able to start developing some predictive trends which would say that given this kind of a pile-up, there is going to be a congestion coming up in next 3 minutes or 5 minutes, and if there is a time, we might be able to actually provide downstream signals which would allow us to change lanes or have people exit and take alternate routes.
To test his theory, Trivedi began deploying omnidirectional cameras around the UCSD campus and overlooking Interstate 5. The goal?
Ubiquitous or all-pervasive video sensing.
He calls them DIVAs, Distributed Interactive Video Arrays. They are connected via the internet to the control room in his lab and through there to each other.
One computer is analyzing what is being seen by the omnidirectional camera feed, and based upon the analysis of that, it can give commands to this other camera to zoom in and give you a much detailed description or view of that event.
One obstacle, how to tell the cars from the road. At noon, it's not hard, but as cars cast shadows, computers have a hard time distinguishing between shadow and car. So graduate students, including Ofer Ackler in the lab, came up with algorithms that allow the computer to distinguish cars from their shadows. The researchers also had to come up with a way to make sense of the warped panoramic feed from omnidirectional cameras.
It's in some sense distorted intentionally because you're trying to look at the whole thing. But then computer vision algorithms or human interpreters, they are not really very comfortable looking at such a crooked, distorted view of the world. So you need to have techniques, algorithms, which would unwarp them.
In his vision of a massively censored road, these cameras would provide pervasive video coverage with the video accessible via high-speed wireless internet into a control center anywhere in the state. So if the cameras alert an operator to a problem, what then? If a motorist runs out of gas or has an accident? For that, Trivedi turned to a mix of robotics and interactive web-based telecommunications. He sees small base stations at regular intervals like emergency phones, especially on the most congested or dangerous stretches of highway. If the cameras detect an incident, the base station would automatically deploy small mobile probes not much bigger than orange hazard cones. Wirelessly controlled, they would be directed to their safety positions automatically based on visual data input from the nearest camera.
These things can automatically get deployed. So you will secure that area so nobody else kind of runs into you while you are seeking help. Also, if you are able to deploy something downstream like a flashing sign or something, then that would also help reduce the speed of the other lanes of traffic.
Trivedi also built a command module. He calls it MIA for Mobile Interactive Avatar that can get to the scene faster than any emergency crew. The mobile system
has cameras on it as well as displays on it, which allows you to have a two-way conversation in a audio-video form from anywhere in the world to any site where the accident needs to be cleared.
By looking close up at the incident, in this case involving researcher Andrew Kosand, and even talking virtually face-to-face with someone on the scene, emergency officials could better gauge what sort of emergency response is needed. A tow truck maybe, or an ambulance, the police, or all of them. Shaving just 5 minutes off the average response time, Trivedi says, could save dozens of lives each year on California roads and relieve congestion much faster.
Eventually, I think that there would be a human being also who is playing a role in saying that what is that I want to deploy, where, and actually be in communication with people who need help.
Even as his team was working on interactive video arrays for roads, Trivedi began working on ways to adapt that technology indoors. It started as an experiment in video conferencing, turning a room into what Trivedi calls an intelligent environment. This room in his lab is equipped with 12 cameras and 8 microphone arrays, all connected to each other. As researcher Kosia Huang enters the room, cameras track his position, and if he is speaking, microphones hand off to each other depending on where he is. The computer can even recognize him if he's been there before. Trivedi quickly realized the potential of adapting such networks inside the car.
Safety of the driver and occupants can be enhanced utilizing these kinds of video networks.
The first safety hazard Trivedi addressed was cell phone use.
Dialing while driving has become second nature, and so have the problems associated with using a cell phone in a car. But this research project at UCSD may soon bring about a change.
The ongoing research at UCSD has already caught the attention of reporters because of the potential benefit. Research shows that using a wireless phone reduces a driver's reaction time by roughly 30%. While hands-free phones, headsets, and other technologies can reduce that risk, they don't do so by much. Looking for a new approach, and with funding from Mercedes' parent company Daimler Chrysler and the University of California's Digital Media Innovation Program, Trivedi began exploring how to turn the cockpit of the car into a mobile intelligent space. Using a structural model of a Mercedes S-Class sedan provided by Daimler Chrysler, researchers outfitted the vehicle with a raft of sensors, omnidirectional cameras, rectilinear cameras, radar, LiDAR, and other sensors.
If we can develop a system which can actually assess the state of the driver as well as the state of the traffic just around that car very reliably and very quickly, then that particular thing can actually be used to allow or modulate the conversation between the driver and whosoever else is trying to talk to them.
Here's the rationale. Today, if you are driving and someone is in the front passenger seat, it's not considered dangerous to be talking. Why? When you are both in the car, the visual context is the same. The passenger knows how relaxed or how tense the driver is, or how difficult traffic conditions are. Based on that, the passenger subconsciously modulates the conversation and maybe stops talking altogether.
All of that kind of a feedback which is associated just because you are sharing the same space is absent when you are on a cellular phone.
To ensure vision at night or in a tunnel, the car also comes with near-infrared illuminators. They emit light humans cannot see, but which is bright enough for a camera to perceive.
We have also added thermal infrared cameras because again during the night the temperature of the driver and the way our faces and the skins look, that doesn't change. So that works quite reliably during dark conditions.
Next comes the hard part, using the cameras inside the car to analyze the state of the driver and his or her environment. Trivedi starts with the face.
We have been developing some new types of computer vision algorithms which allow us to do tracking of individual features on people's faces like lips, eyes, and the way our eyebrows move. And then based upon that we have developed a whole statistical decision theory based classifier which allows us to actually very reliably and very rapidly, like 30 frames per second, recognition of the state of the driver.
Researcher Joel McCall helped develop this smiley face, which reflects his facial features in real time. For now, it mainly tracks the position of the mouth, smiling or frowning, and eyebrows. But McCall says it can be refined to reflect a wider variety of features and moods. Tracking the driver's eyes can also be automated. Tim Schoenmakers has worked on systems to analyze blink rates that may indicate drowsiness or intoxication, or gaze analysis following where the driver is looking.
Difficulties arise when the drivers are not looking straight up front, or if they are looking straight up front but they are getting drowsy. So the blink rates are changing. So can we develop systems which can do this eye blink and eye gaze analysis?
In principle, Trivedi's heavily censored Mercedes could produce multiple streams of video so that an outside caller equipped with the next generation of video-enabled cell phones could see the driver and see what the driver sees. But multiply that data stream by millions and the bandwidth for such a system isn't feasible. Trivedi's solution: boil all the video information that comes in down to a few simple scenarios.
What we would like to feed is really very short, like a 1-bit or 2-bit information which is going to be characterizing the state of the driver, whether they are distracted, whether they are relaxed, whether they are attentive or not, and that could be really added on to the voice channel that you are using on your cell phone, and the cell phone itself can have an extra light which would say that if the light blinks red, then obviously the conditions there are difficult.
Having begun work on assessing the state of the driver, Trivedi's team, with backing from Nissan, in June launched a $3 million, 3-year project to address the problem of driver distraction.
This current year, American roads are going to see 42,000 fatalities. So these are the people who are going to die on the US roads. Out of that, 6,000 deaths are going to be directly related to driver distraction.
Computer vision can help the car identify what's going on in the environment and draw your attention to those situations. To support the driver in a wide number of contexts, you need to have a sensor system including cameras, radar, sonar, GPS, navigation databases, communication between vehicles and environment that can provide the vehicle sufficient information about what's going on around the vehicle.
We would like to have a lot of these technologies serve as our co-pilots, so to say. So they are there, but they are really not distracting us, but they are helping us reduce the distraction and even make us aware of pending emergencies.
Trivedi calls it a human-centered intelligent driving assistance system, a support system really that monitors the driver's state and the state of the road and feeds back information to the driver to maximize attentiveness and safety. The project goes well beyond computer vision.
We must work with psychologists who know how human beings perceive different conditions, how do they switch the priorities of different tasks that they are doing.
Trivedi brought in other UCSD faculty, creating an interdisciplinary team under the auspices of Cal IT Squared, the California Institute for Telecommunications and Information Technology.
Inside Cal IT Square, we actually have a layer which is called Intelligent Transportation and Telematics layer, and we have faculty members in that particular layer who are from psychology, cog science, computer science, supercomputing center, and electrical engineering, mechanical engineering.
The team Trivedi put together includes psychologist Hal Pashler, a leading expert on multitasking. Jim Holland in cognitive sciences works on human-machine interfaces, and Bhaskar Rao, Trivedi's colleague in electrical and computer engineering, deals with signal processing and decision theory. All the sensor information on the state of the driver will be fused with models of the psychology of the driver, and a computer then creates appropriate feedback.
If I'm losing the lane and I'm going to get in the ditch or something, before that maybe my steering wheel will start vibrating a little bit, which is called haptic feedback basically, and that might shake me up and bring my attention to do some corrective action. Or maybe there is something in my seat which might give me a little indication that I need to be a little more attentive. Or there are some lights or alarms that can ring. But what is the optimum way in which you can provide that feedback without really generating a distraction because of the alarm itself?
Consultant Erwin Bohr, an expert on interfaces, says the feedback mechanism has to be intuitive for the driver.
A good interface is one where you look at it and it's immediately obvious what you can do with it. It's called the ecological interface. In driving, that's very important because there is really not much time for cognition to be thinking about what is it exactly that this button facilitates. Assisting the driver in controlling the vehicle, you ideally want to communicate through the control channels, the steering wheel, the pedals, and use what are called ecological sounds. And rather than a beep, perhaps you would show or sound a horn. Rather than show a little light that goes on, perhaps if the car in front of you brakes and you're not looking, you want a red flash in the car. Those are much more intuitive to a driver than perhaps your standard interfaces.
The Nissan consultant says that even as Trivedi's team works on ways to reduce driver distraction, automakers are introducing new in-car technologies, so-called telematics, that could potentially distract them further. Even if voice recognition were 100% accurate, so drivers could keep their eyes on the road and hands on the wheel, cruising the internet while driving could be hazardous.
Say you want to book a movie, schedule a movie, and you call up the movie theater and they give you 7 movies with times. For the driver to keep all that in memory, they're basically internally rehearsing. And in that process, they may lose contact of what's going on in the environment. They're cognitively so loaded that perceptually they may be blinded to some degree.
The project will get underway in earnest in July when Trivedi's lab takes delivery of an Infiniti Q45 sedan. The vehicle will be heavily censored and tested on the open road as researchers write, test, and refine the computer algorithms that could eventually make the smart car a reality.
We are really interested in looking at the entire ecology of driving. So it includes drivers and drivers' interactions with other things.
Erwin Bohr thinks Trivedi is on the right track, installing as many cameras and other sensors as possible to gather the maximum amount of information.
If you can only support the driver, say, in the longitudinal direction, there is a car in front of you, yes or no, but the car has no context, whether there may be a sharp curve coming up, whether there may be a fork in the road, whether there may be children jumping on the road, then that system becomes less helpful to the driver.
Meanwhile, even if interactive video arrays can provide ample information to support the driver by pointing out potentially dangerous situations inside and outside of the car, the big question remains.
How do you communicate that wealth of information to something that can be easily interpreted by the driver?
Trivetti's work on video arrays inside the car also led him to work on a clear and present driving danger. While at least 6,000 lives are saved each year by airbags, some 200 deaths occur directly because of them. The victim may be a small adult, a child, or simply someone leaning too far forward at the time the airbag explodes at a peak 200 miles per hour. With funding from Volkswagen,
we are actually developing a video-based system which extracts the three-dimensional volume of a person who is sitting in the car seat. And we must do that thing very accurately, and the research challenge is we must do that extremely fast.
How fast from the instant of impact to the time the bag is fully inflated is only 25 to 30 milliseconds. That means an onboard computer has to receive video input, assess the size and position of the driver, and decide whether to inflate the airbag partially, fully, or not at all of it in 15 seconds or less. Trivetti thinks it's doable and aims to begin testing the system on the road in late 2002. Trivetti admits that some of the technology he's working on could be controversial, but he thinks the potential benefits in saved lives and less congestion on the roads will be worth any downside that smart cars may present.
Eventually, it is not going to be the technology, whether it is able to do that or not, but we as a society are willing to accept the benefits as well as the costs associated with whatever tool we are going to allow in our cars.
Whether we will ever have fully automated driving out on the roads here, I doubt it. I think we can come very close, but reach a point where the driver can truly just sit back and read the paper is very unlikely.
Mohan Trivetti agrees, but he's convinced that cameras, computers, and the wireless web will make driving safer, and at least 3 global automakers have committed millions of dollars to see if he's right. For UCSD TV, I'm Doug Ramsey.